Welcome to The Deep Dive, the show where we take a stack of critical sources and really just pull out the most important insights, giving you the knowledge you need, fast, but also thoroughly.
Today, we're undertaking a deep dive into, well, the absolute foundation of clinical practice. If you're just starting your journey in medicine, this is your personalized guide. Think of it as a shortcut to mastering the principles that underpin every single prescription you will ever write.
That's right.
We're tackling the core concepts of clinical pharmacology and the rules of good prescribing, all anchored in Chapter 2 of Davidson's Medicine.
Yeah, and our mission today is really tailored for you, the learner, especially if you're feeling that, that crushing weight of information overload. We're not just going to read you the chapter.
Not only.
We're building a mental scaffold. We're going to give you some powerful anchors and hopefully some memorable metaphors. The whole point is to make sure these dense concepts, pharmacodynamics and pharmacokinetics, don't just float away. We want them to stick.
And the need for this foundation has never, ever been greater. The sources we're looking at, they capture this big, complex challenge perfectly. Prescribing is the main tool you have as a doctor.
It's everything.
Right. It's how you restore, preserve, or manage health. But that benefit, it has to be weighed against this constant, challenging triad. The potential for adverse drug reactions, or ADRs, complex drug interactions, and of course the cost.
And what really turns that challenge into a full-blown crisis in some areas is polypharmacy.
Oh, absolutely.
You're just not treating a single condition anymore, especially with our aging population. You're managing patients on 5, 10, sometimes 15 different medications.
Yeah.
The chances of conflict and confusion just skyrocket.
They really do. And to navigate that safely, the modern prescriber has to understand this fundamental duality that really defines pharmacology. We can anchor this using the structure in the sources. On one side, we've got pharmacodynamics, or PD.
Okay.
This is the effect. It's what the drug does to the body, the mechanism, the action.
And then the flip side of that coin is pharmacokinetics, or PK. This is the journey. It's what the body does to the drug.
Exactly. It's the movement. Absorption, distribution, metabolism, and excretion. You'll hear it called ADME. Understanding both of these, PD and PK, it's not optional. It is the absolute foundation. So let's start with the action, the PD.
Okay, let's unpack this. Part 1, pharmacodynamics. This is the molecular level. This is where we define the drug's sort of job description.
Yes, and the mechanism for nearly all modern drugs involves interacting with specific molecular targets. The sources define 4 key categories, and they're all generally proteins.
Right.
Receptors, enzymes, transporter proteins, and ion channels. Think of examples like G protein-coupled receptors or GPCRs, which include the beta-adrenoceptors we'll get into, or something like the insulin receptor.
The classic metaphor here, the one you absolutely have to know, is the lock and key.
Indispensable.
The drug molecule is the key, and that specific molecular target, say a beta receptor, is the lock. And everything hinges on how well that key fits.
And that fit is governed by 2 really crucial terms that you have to separate in your mind: affinity and selectivity.
Okay.
Affinity describes the propensity of the drug to actually bind to the target. It's about the strength of the chemical bond. A drug with high affinity, it sticks quickly and it sticks strongly, even at really low concentrations.
And then there's selectivity. And this is a concept that I think often trips people up.
It does. The source really emphasizes that selectivity is always relative. It's never absolute specificity. No key fits only one lock in the entire universe. It just fits its primary lock much, much better than the others.
That point on relativity feels like a really important clinical anchor.
It's vital. And the best illustration comes from beta-adrenoceptor subtyping. We use beta blockers, they're antagonists, to treat conditions like hypertension or angina. Now, these receptors have subtypes. Beta-1 is mostly in the heart, and beta-2 is mostly in the smooth muscle of the lungs.
So a cardioselective beta blocker is designed to target the beta-1 receptors in the heart. That's where we want the effect. But wait, if selectivity is only relative?
Exactly. Even a highly cardioselective drug still has some residual affinity for the beta-2 receptors in the lungs. Now, that little trace effect is often clinically insignificant in a healthy patient.
But in an asthmatic patient?
Catastrophic, because even mild beta-2 antagonism can trigger life-threatening bronchospasm.
Wow. So the immediate clinical takeaway here is that you must never prescribe a beta blocker to a patient with severe, uncontrolled asthma. Not even if it's marketed as cardioselective. Selectivity failure can kill.
That's your anchor. Selectivity is always a balancing act, never an on-off switch.
Okay, so moving on to the action itself. Once the drug binds, it either mimics the body's natural stuff or it blocks it. agonists and antagonists.
Right. An agonist binds and produces a conformational change, which leads to a biological response. But there's nuance here. A full agonist produces the absolute maximal response that system is capable of.
And then you've got the partial agonist.
The partial agonist is interesting. It binds, it activates the receptor, but, and this is the key part, it cannot produce that maximal signaling effect, even if every single receptor in the body is occupied by it. It just sits at a lower level of activity.
Okay, let's use a metaphor. If receptor activation is like a skyscraper you're climbing, a full agonist lets you take the elevator all the way to the top floor.
Right to the top.
The partial agonist is like having a key that only lets you get to the 10th floor. No matter how many keys you use, you can't get past that ceiling.
That's a perfect way to think about it. Now, the antagonist is the key that fits in the lock, but just jams it. It doesn't cause any signal itself. Its only job is to stop the natural agonist, or another drug, from binding and acting.
And antagonists have a couple of different ways they can block things, right?
They do. A competitive antagonist fights for the exact same parking spot as the agonist. Its effect can be overcome if you just increase the concentration of the agonist. It's a numbers game.
And the other type?
A non-competitive antagonist. This one doesn't fight for the same spot. It might bind irreversibly somewhere else, or maybe it acts downstream in the signaling pathway. But the bottom line is, its effect can't be overcome just by adding more agonist.
Okay, so we know how they bind and act. Now we have to measure the effect. Dose-response relationships and safety. This is where we start comparing drugs.
When you plot the dose against the response, you get this typical S-shaped or sigmoidal curve. And that curve immediately tells you two essential things about a drug: its efficacy and its potency.
Let's really hammer home the most important anchor in this whole section. Efficacy is the height of the skyscraper. It's the Emax.
Yes, it's the maximal response you can possibly achieve. It's the absolute ceiling of the therapeutic effect, no matter how high you push the dose.
And this is clinically huge, right? Because it lets you compare drugs that work in totally different ways.
Totally. For instance, in treating acute volume overload, loop diuretics have far greater therapeutic efficacy. They achieve a much higher skyscraper in terms of fluid removal than thiazide diuretics do. No matter how much thiazide you give, you just cannot match the sheer power of a loop diuretic.
And potency, then, is just how much fuel you need to start climbing that skyscraper.
Exactly. Potency is measured by the ED50, the dose you need to get to half of that maximal response. So a more potent drug has a lower ED50. You need less of it.
Okay, here's a classic nuance test question. If drug A is 10 times more potent than drug B, does that mean drug A is a better drug?
Absolutely not. And this is a key point. Potency just determines the dose size on the label, 10 milligrams versus 100. A less potent drug can be just as good or even better if you can just increase the dose to reach that maximal efficacy. You can't increase efficacy beyond its Emax, no matter how potent it is.
Which leads us straight to the concept that really determines a drug's usefulness in the real world: the safety net or the therapeutic index.
The TI. It's the ratio of the dose that causes adverse effects to the dose that gives you the beneficial effects.
So back to our metaphor. The therapeutic index is the distance between the effective dose highway and the toxic effects cliff.
I love that. The wider that gap, the more forgiving and therefore the safer the drug is for everyone.
And for most drugs, that gap is pretty wide.
It is, thankfully. Most have a TI over 100. You can be a bit inexact with the dose, and the patient will be fine. However, you must commit to memory of the drugs with a narrow, dangerous therapeutic window, a TI of less than 10.
These are the low TI classics: digoxin, warfarin, insulin, phenytoin, and opioids.
Let's just talk about the clinical implication here for a second. Using warfarin. Warfarin prevents clotting. That's the therapeutic effect.
Yeah.
But if you accidentally increase the dose just a tiny bit above that range, you fall right off the cliff into severe bleeding.
So for these specific drugs, dose titration isn't just careful, it's meticulous. It requires constant monitoring, often with blood tests, because that cliff edge is always just right there.
It's always dangerously close, and that's why these drugs are so often involved in medication errors.
Okay, let's circle back to our check-in question now that we have this deeper understanding.
Check in Q1, the nuance test. If you're comparing two drugs and drug A is 10 times more potent than drug B, is drug A automatically the better choice?
And if you're thinking not necessarily, you're on the right track. The choice depends on their maximal efficacy, the height, and most critically, the therapeutic index, that distance to the cliff.
Perfect. Now, the relationship between dose and response isn't static. The body fights back, which can lead to a loss of response. We call this generally desensitization.
And this happens on two different timelines.
Right. The first is incredibly rapid, tachyphylaxis. This desensitization can happen very, very quickly, sometimes even after the first dose.
The analogy for this one has to be the sprint runner who instantly runs out of stored energy.
That's it. The cause is usually rapid depletion of some critical chemical, like a neurotransmitter, or the receptor itself just gets shut down chemically.
The second, slower response loss, is tolerance.
This develops gradually, over days or weeks. Here, the body has time to adapt. Maybe it reduces the actual number of receptors available, or it kicks in some counter-regulatory systems. For example, if you give a vasodilator, the body tries to compensate by retaining salt and water, which kind of offsets the effect over time.
And when you abruptly stop a drug that's caused the body to adapt like that, you get a serious withdrawal effect or a rebound.
This is a major safety issue. The sources list some high-risk examples. If you stop an SSRI antidepressant abruptly, you can get severe dizziness, agitation, stop benzodiazepines or alcohol suddenly, and you risk seizures.
And perhaps the most high-stakes example is glucocorticoids, steroids.
Oh, yes. Long-term steroid use suppresses the body's own steroid production system. Stopping them cold turkey can lead to life-threatening adrenal insufficiency. That's why these drugs require a slow, deliberate taper.
Okay, so let's try to summarize part one.
Key takeaway for Part 1, pharmacodynamics. Pharmacodynamics defines the drug's intrinsic properties. It sets the maximum possible effectiveness, which is efficacy, and the inherent safety margin, the therapeutic index. Mastering this helps you choose the right drug.
We've established the drug's potential. Now we get into the messy, variable reality of its journey through a human body. This is pharmacokinetics, ADME. This is what determines the practicalities of dosing.
Exactly. Understanding PK is how you choose the right route, the right dose, the right frequency. It also explains why patients vary so much in their response to the exact same dose.
Let's start with absorption getting into the system. The most common route is oral. It's convenient for the patient, but it's a nightmare for the drug.
Yes. Our anchor here has to be. The oral route is an obstacle course. It's challenging because the drug faces at least 4 major hurdles before it even sees the systemic circulation.
Okay, so first, it has to survive the stomach acid.
Right. Second, it can't bind irreversibly to food in the gut. Third, it has to be able to pass through the gut wall. But the biggest hurdle of all is the fourth one. First-pass metabolism.
The liver.
The liver. This pre-systemic clearance happening in the gut wall, and especially the liver, means a huge chunk of the dose can be metabolized before it ever gets into the general circulation.
So if a drug has significant first-pass metabolism, we end up with low bioavailability, which is the proportion of the dose that actually makes it into the circulation intact.
That's why drugs like morphine or propranolol require a much, much higher oral dose than an intravenous dose to get the same effect. The body is just really good at getting rid of it on that first pass.
So when we need certainty, and we need it fast, we bypass the gut entirely with parenteral routes.
4C, intravenous is the gold standard for certainty. 100% bioavailability, immediate effect. It's what you need for a critically ill patient. IM, intramuscular, and SC, subcutaneous, are good for self-administration, like insulin, but the absorption can be a bit less predictable.
Okay, so once it's absorbed, the drug is in the blood, and it's ready for distribution. Where does it go?
Well, this is governed by its lipid solubility and, critically, how much it binds to plasmaproteins, mainly albumin. These proteins act like sponges, soaking up the drug and keeping it trapped in the blood. Only the free, unbound drug can leave the blood and have an effect.
This brings us to that tricky concept, the volume of distribution, or V. That's not a literal volume, is it?
No, it's an apparent volume. It's a calculation. But our anchor helps. V is the size of the bucket the drug is poured into. A small bucket means the drug dose stays concentrated in the blood, giving you a high initial plasma concentration.
So drugs that are highly protein-bound, like warfarin or aspirin, they stay in the blood. That's a small bucket, a V of less than 10 liters.
Right, but then you have these highly lipid-soluble drugs, like the low-TI classic digoxin. These molecules are masters of tissue binding. They leave the blood really fast and just sequester themselves in fat and muscle tissue.
So the concentration in the blood drops like a stone.
It plummets. And that gives you the apparent calculation of a massive bucket, sometimes over 100 liters.
And what's the clinical significance of that? If a drug is stuck in that huge tissue bucket,
Then Even if your kidneys are clearing plasma really fast, the drug just keeps leaching back out of the tissue stores into the plasma to be cleared.
Which means?
It means drugs with a large VEED have significantly longer half-lives. It explains why digoxin has a 36-hour half-life and takes days and days to reach a steady state. It's hiding out in the tissues.
Now the body starts getting ready to eliminate the drug. Metabolism and excretion.
This is the detoxification engine, mostly in the liver. Its whole job is to convert lipid-soluble, absorbable drugs into water-soluble, excretable metabolites. It's the only way the kidneys can effectively get rid of them.
And we often hear about the two metabolic steps, phase 1 and phase 2.
Phase 1 is the initial chemical modification, oxidation, reduction, that kind of thing. The stars here are the cytochrome P450 or CYP family of liver enzymes. They basically stick a chemical handle on the molecule.
To get it ready for phase 2.
Exactly. Phase 2 is conjugation. That phase I metabolite is now linked to an endogenous substance, like glucuronide, to create a highly water-soluble molecule. This is non-negotiable for renal excretion. If a drug is still lipid-soluble when it hits the kidney, it'll just diffuse right back into the bloodstream.
Here's a practical point that's often missed. Phase I metabolism, the CYP system, is much more sensitive to things like aging and liver disease than phase 2 is.
That's right. So a drug that relies mainly on phase 2 conjugation is generally a safer choice in an elderly or liver-impaired patient. It gives you a better safety margin.
We mentioned prodrugs earlier. These are inactive molecules that need metabolism to become active.
Right, and a critical safety reminder here, the antiplatelet drug clopidogrel is inactive until it's activated by liver enzymes. If a patient is a poor metabolizer because of their genetics, they won't activate the drug. They'll be at risk of clots, even though they're taking the pill every day.
Finally, we get to excretion. Most drugs leave via the kidneys, the renal route.
Correct. But the kidney is complex. Weakly acidic drugs, like salicylates, can be reabsorbed from the tubules. So in an overdose, we can actually manipulate the urine pH, make it alkaline, to trap the drug in the filtrate and speed up its excretion. It's a life-saving PK intervention.
And we can't forget the recycling loop and enterohepatic circulation.
This loop prolongs a drug's life in the body. The drug gets excreted into the bile, dumped into the gut, and then gut enzymes can sometimes cleave it, releasing the lipid-soluble drug to be reabsorbed all over again. It just goes round and round. Digoxin and morphine are classic examples.
All of this, the whole journey, culminates in determining the dosage schedule, which is based on elimination kinetics.
And the standard measurement is clearance. But thankfully, the vast majority of drugs follow predictable first-order kinetics.
Which means the rate of elimination is proportional to the concentration. A constant fraction of the drug is cleared in a given time.
That's it. So if you have 100 milligrams and the half-life is 2 hours, you clear 50 milligrams. And you have 50 left, so in the next 2 hours, you clear 25. The decline is exponential, and the half-life is constant. It's predictable. If you double the dose, you double the concentration.
Now for the high-stakes exception, zero-order kinetics.
This is a life-threatening concept you must master. In zero-order kinetics, the elimination capacity, the liver enzymes, gets saturated. The system gets overwhelmed, even within the therapeutic dose range.
So the body switches from clearing a constant fraction to clearing a constant amount.
Yes, and the consequences are dire. If the rate of administration is more than that saturated clearance rate, the drug concentration just accumulates progressively. It goes up and up and up. The half-life is no longer constant. The classic dangerous examples are phenytoin and alcohol.
Let's run a scenario on that because it's so important. A patient is on a standard dose of phenytoin, a zero-order drug. A doctor increases the dose by just a little bit, say 10%.
With a first-order drug, a 10% dose increase gives you a 10% concentration increase. Predictable. But for phenytoin, that small increase might push the total amount just over the saturated clearance capacity.
And then?
The drug begins to accumulate uncontrollably. It leads rapidly to severe toxicity. The change is nonlinear, and it's disastrous.
That distinction between constant fraction and constant amount is just, it's the difference between safety and severe toxicity.
Right. And for continuous therapy, the goal is to achieve steady state. You want to keep the drug concentration in that therapeutic window.
Which happens when the amount coming in equals the amount going out.
And here's the essential time anchor you need to remember. Steady state is like filling a leaky tub. It takes approximately 5 half-lives to stabilize the water level.
That math is so critical. If a drug has a 12-hour half-life, you reach steady state in 60 hours. If you start digoxin with its 36-hour half-life, you're waiting over a week for that steady concentration.
And that's often too long if the patient is acutely ill. That delay is why we use a loading dose. The loading dose is calculated to rapidly hit that steady state plateau, which you can then maintain with the smaller regular doses.
We also have to think about dose intervals. Dosing 4 times a day is a pain, and adherence plummets.
Which is where modified release, or MR formulations, come in. They slow down absorption, smoothing out the peaks and troughs. This is crucial for low TI drugs like levodopa for Parkinson's.
Okay, let's run the second check-in, focusing on that dangerous difference between first and zero order.
Check in Q2, the saturation test. You have 2 patients. One is taking amoxicillin, which is first order. The other is taking phenytoin, zero order. If you increase the dose for both by 20%, what happens?
If you correctly identify that the amoxicillin concentration increases predictably by 20%, but the phenytoin patient risks uncontrolled progressive accumulation and toxicity, then you've absolutely got it.
Key takeaway for Part 2, pharmacokinetics. PK determines how and how often you dose a drug. You have to understand half-life, VD, and critically distinguish between linear, first order, and nonlinear, zero order kinetics to dose safely.
Okay, we've covered the predictable factors, sort of the averages. Now we enter the real world, where patients are highly variable and the risk of harm is always there. This is Part 3, acknowledging individual variation and managing adverse outcomes.
Good prescribing is all about anticipation. You have to adjust dosing based on so many patient factors. Age, organ function, body weight. For instance, obesity significantly increases the VD for lipid-soluble drugs, so a standard dose might be completely ineffective.
And the real frontier of this is pharmacogenetics. We now know that simple genetic differences explain why some patients metabolize drugs fast and some slow, and why others have these bizarre, unpredictable reactions.
The CYP450 enzymes are the best example. Genetic variations in the CYP2D6 enzyme mean people are poor metabolizers, normal, or even ultra-rapid metabolizers.
And how does that actually play out in therapy?
Well, take the prodrug codeine. It has to be metabolized by CYP2D sulks into morphine to work. A poor metabolizer gets almost no pain relief because they can't activate the drug.
And an ultra-rapid metabolizer.
They convert it so quickly, they risk opioid toxicity from dangerously high morphine levels. This kind of genetic testing is becoming standard for certain high-risk drugs.
We also see genetic variation on the pharmacodynamic side.
We do. G6PD deficiency is a classic. Patients with this lack a key enzyme in their red blood cells. If you prescribe certain oxidant drugs, they risk severe sudden hemolytic anemia because their red cells are just too fragile.
And then there are the really high-stakes genetic reactions, the hypersensitivity ones.
Yes, the HLA polymorphisms. Specific HLA types are strongly linked to severe life-threatening rashes when you prescribe certain drugs, like the HIV drug abacavir. Screening for the specific HLA type is now mandatory before starting that therapy.
Okay, let's switch gears to the inevitable, adverse outcomes. We need to be crystal clear on the definitions here. ADR, side effect, toxicity.
Right. An adverse drug reaction, an ADR, is generally an unwanted or harmful reaction that happens under normal use. Usually means you have to stop the drug or lower the dose. A side effect is a non-intended effect, often predictable, like a dry mouth, that happens within the normal therapeutic range.
And drug toxicity is specifically the bad stuff that happens because the dose or concentration has gone above the therapeutic range.
The prevalence of ADRs is it's a public health crisis. The sources say they account for around 7% of all emergency hospital admissions. And the really sobering statistic is that more than half of these ADRs are considered avoidable. Better prescribing could prevent thousands of hospitalizations.
To reduce that harm, we have to classify the risks. Let's start with the standard A-B classification.
Type A, augmented ADRs are the low-hanging fruit. They're predictable, they're dose-dependent, and they're common. They're just an augmented version of the drug's known pharmacology. Constipation from opioids is a perfect type A. You can manage them by reducing the dose.
So if type A is predictable, type B is the opposite.
Type B bizarre ADRs are the terrifying ones. They're unpredictable, not related to the drug's core action, they're not really dose-dependent, and thankfully they're rare. They're often due to some kind of immunological or genetic hypersusceptibility.
So penicillin anaphylaxis is the classic type B.
The classic, or those severe skin reactions linked to HLA polymorphisms.
The sources also expand this with the DOTS classification, which is about timing.
Right. DOTS helps capture things missed by A and B. Type C, chronic reactions need long-term exposure, like osteoporosis from long-term steroid use. Type D delayed reactions surface long after the drug is stopped, which makes them incredibly hard to link.
And type E, end of treatment, are the withdrawal effects we talked about earlier.
Exactly.
Okay, let's go a bit deeper into hypersensitivity reactions because they represent the most acute type B reactions.
These are immunological responses. Type I is that immediate IgE-mediated life-threatening anaphylaxis, penicillin, bee stings. Then you have type IV reactions, which are T-cell mediated and take days or even weeks to develop.
And these type IV reactions include some really severe stuff, like Stevens-Johnson syndrome, SJS.
Absolutely. SJS and the even worse version, TEN, involve widespread blistering and sloughing of the skin. They are medical emergencies with high mortality rates, and they're typically linked to drugs like carbamazepine. This is a classic type B, type D bizarre delayed reaction.
Yeah, let's just make it even more complex. Drug interactions.
These are most likely when you have a low TI drug in the mix or one with saturable metabolism. And the interactions can be either pharmacodynamic at the site of action or pharmacokinetic, messing with the drug's concentration.
PD interactions seem kind of intuitive.
They often are. Alcohol and benzodiazepines together cause a massive increase in CNS depression. That's a synergistic PD interaction, or antagonism. Giving a beta agonist like salbutamol with a beta antagonist like atenolol means they fight for the same receptor, and the salbutamol just doesn't work as well.
PK interactions, on the other hand, are less intuitive but just as dangerous. They almost always involve that CYP450 system in the liver.
And we have two villains here. CYP enzyme inducers and CYP enzyme inhibitors.
Okay, inducers.
Inducers, like rifampicin or St. John's wort, they ramp up the production of CYP enzymes. So if a patient is taking another drug that's cleared by that enzyme, it suddenly gets cleared way too quickly. The plasma concentration drops and you get therapeutic failure.
And inhibitors.
Inhibitors are faster and often more dangerous. They physically block the existing enzymes, so the clearance pathway is suddenly blocked, and the drug concentration spikes upwards fast. This is a huge risk with a low TI drug like warfarin.
Right, so you add an inhibitor like clarithromycin, the warfarin level shoots up, and the patient starts bleeding.
Precisely.
Finally, we have to confront the reality of medication errors. These are avoidable events, and they're really common, especially in the chaos of a hospital ward.
Very common, and often it's junior doctors. The errors include dosing mistakes, poor transcription, and critically, failures of medicine's reconciliation, forgetting to continue or restart a patient's regular meds when they're admitted or discharged.
When we analyze these errors, human error theory makes a distinction between slips and lapses on one hand and mistakes on the other. Why is that distinction so important?
Because it tells you how to fix the problem. Slips and lapses are unintended actions. The doctor knows the right dose, but they're distracted or rushed and they write mg instead of micrograms.
So the error is systemic. The prevention needs to be systemic.
Exactly. You need checking routines, better computer systems, things that reduce distraction. But a mistake is deeper. A mistake is an intended action based on flawed knowledge. The prescriber thinks they're doing the right thing.
Like prescribing a beta blocker to an asthmatic.
Perfect example. You can't fix a knowledge gap with a checklist. The prevention there is training, education, and strengthening the knowledge base.
And when an error is discovered, the response has to be clear. Patient safety first, record it, inform everyone, and report it in a no-blame culture so the system can learn.
Yes, and that reporting system for rare ADRs is called pharmacovigilance. It's the only way we ever find out about these rare type B reactions that don't show up in the initial trials.
And that's where the trend analysis comes in.
Right. It's a methodical way to assess a potential ADR, temporal relationship, re-challenge, exclusion, novelty, de-challenge. It turns a suspicion into evidence.
Okay, let's wrap this part up.
Key takeaway for part 3, adverse outcomes. ADRs and errors are frequent and often avoidable. You have to anticipate them by understanding genetics, and you must be able to distinguish between predictable type A and bizarre type B reactions to minimize risk.
We've covered the science, the risks. Now we put it all together into the structured, systematic process of good prescribing in practice.
And it all starts with mutual agreement. You establish the therapeutic goals, but critically, these goals have to be shared with the patient. That's concordance. If a patient doesn't understand why they're taking a drug, adherence will be terrible.
When it comes to choosing the drug, it's a careful balance of efficacy, safety, cost, and especially coexisting disease.
Coexisting disease is the main driver of complexity. You know, a patient needs an anti-inflammatory. NSAIDs are great, but if they have a history of peptic ulcers, NSAIDs are out. You have to choose an alternative, constantly balancing the pros and cons.
When setting the dose regimen, the default setting is always caution.
The golden rule is start low and titrate slowly upwards. This is mandatory for the elderly, anyone with renal or hepatic problems, and for any low TI drug. The only exceptions are when you need an immediate effect, like with antibiotics, or when you're using a loading dose.
And the importance of timing goes beyond just convenience.
Oh, absolutely. Diuretics cause urination, so they have to be given in the morning. Some statins are best given at night because that's when the body's cholesterol production is highest. It's about optimizing efficacy and safety.
What about the generic versus brand name debate for formulation?
Standard practice is to prescribe by the generic name to save money. However, you must prescribe by the specific brand name for those few low TI drugs, where even tiny differences in manufacturing between generics could be dangerous. Lithium and phenytoin are the two absolute classics where you always stick to the same brand.
Okay, let's dive deeper into the fragile patients, starting with renal disease.
Kidney impairment is common, and if a drug is cleared by the kidneys, the dose must be reduced. We use the eGFR to guide this. High-caution drugs include digoxin, lithium, and aminoglycosides. And remember, renal impairment doesn't just affect clearance, it can also increase sensitivity to a drug's effects.
And what about hepatic disease?
The liver has a huge reserve capacity. You usually only need to modify doses in advanced liver failure, but unlike with the kidneys, we don't have a single good test to measure liver function for drug metabolism, so you have to be guided by clinical signs.
High caution drugs for liver disease.
Warfarin is a big one because the liver makes clotting factors, also opioids and diazepam.
And the largest group of fragile patients, older patients.
They get hit with that lethal trifecta. Reduced drug elimination, increased sensitivity to drug effects like confusion and falls, and of course polypharmacy, which just exponentially increases the risk of interactions. The approach has to be so conservative, lower doses, slower titration, and constantly trying to simplify their regimen.
Now we get to the critical moment, writing the prescription. The sources stress absolute total meticulousness on the hospital drug chart.
Yes. Patient details must be complete, and the section for allergies and previous ADRs must be checked and completed before you even think about writing a single drug name.
Let's just hammer this home. The rules on units and abbreviations are non-negotiable for safety.
To prevent those thousand-fold errors, you must always write micrograms in full, never use the abbreviation, and you must always write units in full, never use the letter U. This simple rule prevents so many catastrophic errors.
And what are the key procedural safety steps for a junior doctor, especially in those high-risk moments?
First, never ever rush. And second, always ask a colleague to independently check any drug dose calculation that relies on patient factors, like weight or renal function. Double checking is your best defense against slips.
Prescribing is also about collaboration, about ensuring adherence.
Non-adherence is rampant, especially with preventative drugs where the patient doesn't feel any different. The solution is communication. Shared decision-making, explaining the reason for the therapy, and establishing follow-up.
Finally, we monitor the experiment, monitoring drug therapy.
We prefer to measure the clinical endpoint, the actual effect we want, like the INR for warfarin. But sometimes that's hard, so we use a surrogate endpoint, like cholesterol level as a surrogate for future heart attack risk.
When do we escalate to measuring the actual drug concentration in the blood therapeutic drug monitoring, or TDM?
TDM is expensive, so it's only justified if 3 criteria are met. 1, the clinical outcome is hard to monitor. 2, there's a predictable relationship between concentration and effect. And 3, critically, the drug has a low therapeutic index.
So the usual suspects again: digoxin, lithium, phenytoin.
Those are the ones that justify TDM. And remember the rules of timing. You take the sample at the trough right before the next dose, and only after you've confirmed that steady state, those 5 half-lives, has been reached.
We also have to remember that for protein-bound drugs, the total concentration can be misleading if the patient has low protein levels.
In that case, more of the drug is free and active, so toxicity can happen even when the total level looks like it's in the target range. It's a big issue with phenytoin in malnourished patients.
Okay, let's summarize Part 4.
Key takeaway for Part 4, practice. Good prescribing requires meticulous attention to detail in high-risk groups and constant monitoring. Mastering TDM criteria and sample timing is essential for managing low TI drugs safely.
Okay, moving beyond the clinic, let's briefly step back and look at the huge regulated infrastructure that delivers these therapies. Drug development is just an astronomical gamble.
It really is. The sources highlight the reality. It takes over a decade and costs upwards of $2 billion per drug. That investment justifies the patent protection manufacturers get, and the development is defined by 3 clinical phases.
Phase 1 is the first step into humans.
Right. Small trials, healthy volunteers. The questions are about basic safety and PK/PD. How is it absorbed? How is it eliminated?
Phase 2 shifts to the actual patients.
Small groups of patients. The focus is on effectiveness, does the concept work, and finding the right dose range.
And Phase 3 is the big one.
Large, randomized controlled trials with hundreds or thousands of patients. This is to confirm efficacy and to detect the more common ADRs. Only after a successful Phase 3 can a company even apply for regulatory approval.
The regulators like the FDA or MHRA grant market authorization, and they produce that essential document, the Summary of Product Characteristics, or SPC.
The SPC is the prescribing Bible. It lists the approved indications, dosage, known ADRs, everything. You have to adhere to the SPC unless you have a very strong clinical justification.
The major challenge in modern medicine is the rise of biologics. These are fundamentally different from traditional drugs.
They are. Biologics are large, complex molecules, monoclonal antibodies, proteins made using biological systems. They can be revolutionary, but they're complex.
And what are their unique safety challenges?
Well, cost is one. But clinically, they introduce new risks like immunogenicity, where the body attacks the drug itself, and a significantly increased risk of severe infection because they often target key parts of the immune system.
Let's just clarify off-label prescribing.
That's prescribing outside the terms of the SPC for an unapproved age group or indication. It's common and often necessary, but the prescriber assumes full responsibility and must get informed consent from the patient.
The final unavoidable layer here is economics. Even after a drug is proven safe and effective, it still has to pass a test of value.
This is a crucial distinction. Regulators confirm safety and efficacy. They do not assess cost effectiveness. That's a separate process, using cost effectiveness analysis, or CEA.
And CEA relies on that measure of benefit known as the Quality Adjusted Life Year, or QELY.
A QAY values a year of life based on its quality. CEA then calculates the incremental cost effectiveness ratio, the ICER. It's the extra cost divided by the QALYs you gain.
The example in the sources is really stark. A therapy costs an extra £16,000 and gives 0.4 extra QALY. That's an ICER of £40,000 per QALY.
And if the national health system has a maximum threshold of, say, £30,000 per QALY, then that therapy, despite being effective and safe, will be rejected for funding. This creates huge economic and ethical friction.
When you're evaluating the data for these decisions, you have to stay critical. Don't be blinded by relative figures.
Always prioritize the absolute risk reduction over the relative risk reduction. A 50% relative risk reduction sounds amazing, but if the absolute risk only dropped from 2% to 1%, the clinical impact is modest. This is captured by the number needed to treat, or NNT. In that case, you'd have to treat 100 patients for just one to benefit.
That was the complete deep dive into clinical therapeutics. We moved from the molecular level with pharmacodynamics, defining the height of the skyscraper efficacy and the distance to the cliff, therapeutic index.
We tracked the drug's messy journey with pharmacokinetics, mapping the oral obstacle course of first-pass metabolism, measuring the size of the bucket with VD, and calculating that life-saving time frame of the 5 half-lives to stabilize the leaky tub.
We confronted the unavoidable risks, the predictable type A ADRs, the bizarre type B reactions, and the high-stakes consequence of zero-order kinetics, where a tiny dose change can lead to disaster.
And we brought it all back to the clinic, prescribing meticulously, reducing doses for the fragile, using those mandatory safety checks like writing micrograms in full, and confirming the need for TDM only for those low TI exceptions.
Remember, every prescription you write is an experiment you conduct on that individual patient. Your initial goal is to master the predictable factors, the PK and PD, so that you can safely anticipate and manage all the unpredictable variations.
And as you grow into your role as a gatekeeper of these powerful chemical tools, always consider this final provocative thought. We accept that regulators confirm a drug's safety and efficacy, but they do not assess its cost-effectiveness. In a world of finite healthcare resources, which ethical principle should guide a committee when deciding whether to fund an expensive, life-extending drug whose cost far exceeds the accepted economic threshold? Mastering that tension is what separates a technician from a thoughtful clinician. Good luck with the experiment.